Good day, everyone. I am Paula Joy Aliposa, and today I will be sharing with you two microbes of industrial importance. The first industrially important microbe I will be sharing with you is Phaeodactylum tricornutum. It is a marine unicellular microalgae which has gained popularity in recent years since it produces a wide variety of products, including eicosapentaenoic acid, fucoxanthin, neutral lipids, chrysolaminarin, triterpenoids, and bioplastic precursors. In fact, due to its significance, it has become the second diatom for which a whole genome sequence has already been generated. Since Phaeodactylum tricornutum produces numerous compounds of interest, it has been harvested, mass-produced, and widely used in various industries. It has been reported by Camacho and others in 2019 that its powdered form has been used in the food industry to improve color, stability, and texture of cookies and biscuits due to the bioactive compounds it contains, including proteins, EPA, DHA, omega-3, and astaxanthin, which also provide an added health benefit, specifically that it possesses antioxidant properties. Not only that, but in capsule form, it has also been used as a food supplement, specifically as an alternative protein source. It has also been used in the agricultural industry as a prebiotic in the feed of animals. The freeze-dried form of Phaeodactylum tricornutum has been used as a prebiotic for the probiotic Bacillus subtilis to improve the immune system and increase intestinal absorption ability of the gilt-head sea bream, or scientifically known as Spars orata. It has also been used to feed other fish larvae and mollusks. Gallas and others also reported recently that Phaeodactylum tricornutum has been recently used for biotechnological applications such as the production of biopharmaceuticals, including monoclonal antibodies. Moreover, engineered Phaeodactylum tricornutum is able to produce recombinant human anti-Marburg virus MABS and functionally glycosylated human anti-hepatitis B MABS. In line with the updated taxonomic classification of the Integrated Taxonomic Information System, Phaeodactylum tricornutum belongs to the domain Eukarya, kingdom Chromista, phylum Basilarhiophyta, class Basilarhiophyceae, order Naviculales, and family Phaeodactylaceae. The pennate diatom Phaeodactylum tricornutum is actually atypical as it occurs naturally in three distinct morphotypes, which are fusiform, triradiate, and oval, as seen in the following slide. However, the fusiform morphotype is the more frequent one. As I mentioned earlier, Phaeodactylum tricornutum is commonly found in marine ecosystems, but is also widespread in brackish waters worldwide. Reports have also indicated finding Phaeodactylum tricornutum in unstable environments, including estuaries and rock pools. In fact, certain environmental conditions have been reported to induce the transformation between the different morphotypes I mentioned earlier. Now that we are familiar with possible sampling locations of Phaeodactylum tricornutum, we can now proceed to collecting samples for propagation. In this protocol I will be sharing from the Woods Hole Oceanographic Institution, P. tricornutum was collected from oceanic surface water from the Sargasso Sea. After securing the samples in appropriate containers, it was then transported to the laboratory. Once the samples have been safely transported to the laboratory, it was subjected to pre-filtration, pre-cleaning, ultrafiltration, and finally sterile filtration. In this protocol by WHOI, an initial 1-liter seed culture was prepared in the lab to condition cells by inoculating 1 liter of F2 media with 5 ml of a growing P. tricornutum culture. Cells were monitored for growth using a Palmer-Maloney counting chamber at 40 times magnification and a Zeiss light microscope. After 5 days of growth, 800 ml of the seed culture was transferred to a fresh 19 liter of F2 media and incubated under the same conditions. An alternative method described in a paper by Shen and others in 2009 also described the usage of horizontal airlift tubular photobioreactors for culturing and mass-producing Phaeodactylum tricornutum in an outdoor setting. In case you were wondering, the F2 media is a common and widely used general enriched seawater medium designed for growing coastal marine algae, especially diatoms. After 7 days of growth with periodic gentle mixing, cells reached stationary phase and the culture was harvested to remove cell biomass and extract extracellular DOC material. Cells were removed by centrifugation at 7,000 rpm for 15 minutes, followed by 0.1 micrometer filtration. Cell pellets from the centrifugation were combined and frozen for future chemical analysis. The 0.1 micrometer filtrate was then frozen until solid phase extraction of DOC compounds. The filtrate can then be subjected to analysis through other chemical tests, and then biochemical methods of extraction can be employed to obtain wanted compounds. The second industrially important microbe I will be sharing with you is Noctiluca scintillans. In line with the updated taxonomic classification of the Integrated Taxonomic Information System, Noctiluca scintillans belongs to the domain Eukarya, kingdom Chromista, phylum Pyrophycophyta, class Dinophyceae, order Noctilucales, and family Noctilucaceae. Noctiluca scintillans is basically an unarmored, free-living, non-parasitic, marine-dwelling, phagotrophic and planktonic dinoflagellate which feeds on phytoplankton, other protozoans, detritus, and fish eggs. What makes it interesting for most people is that in some parts of the world, it is capable of exhibiting bioluminescence. Its bioluminescence is precisely how it got its name, which roughly translates to the sparkling night light, as it gives the night tide a scintillating electric blue appearance. Su and others also reported that in areas where it is abundant, its bioluminescence acts as a sensitive expressive character which provides an indication of its spatial distribution. Moreover, other studies have indicated that this property has a potential to be used in biomedical researches and applications. Although N-scintillans is non-parasitic as I mentioned earlier, it can still be dangerous when ingested by humans who are immunocompromised, particularly individuals with liver problems as it secretes small amounts of ammonia as a metabolic byproduct which may result in ammonia toxicity, hallucinations, metabolic failure, and death. In line with this, it is actually one of the most important and abundant red tide organisms, which makes it an important protozoan species, specifically due to its role in marine food webs in which it is a predator and yet still not at the top of the planktonic food web. Toxic blooms of N-scintillans have been linked to massive fish and marine invertebrate kills. Although this species does not produce a toxin, it has been found to accumulate toxic levels of ammonia, which is then excreted into surrounding waters, possibly acting as the killing agent in blooms. Extensive toxic blooms have actually been reported off the east and west coasts of India, where it has been implicated in the decline of fisheries. Noctiluca scintillans actually occurs in two forms, namely the red and the green morphotype. Red Noctiluca is heterotrophic and acts as a microzooplankton grazer in the food web, whereas green Noctiluca contains the photosynthetic symbiont Pedinomonas noctilucae, but it also feeds on other plankton when the food supply is abundant. Noctiluca scintillans has a worldwide or cosmopolitan distribution in both cold and warm waters. However, there are certain variations in the distribution of the green and red morphotypes. Red Noctiluca occurs widely in the temperate to subtropical coastal regions of the world. It occurs over a wide temperature range of about 10 to 25 degrees Celsius and at higher salinities and generally not in estuaries. It is particularly abundant in high productivity areas such as upwelling or eutrophic areas where diatoms dominate since they are its preferred food source. Green Noctiluca is much more restricted to a temperature range of 25 to 30 degrees Celsius and occurs mainly in the tropical waters of Southeast Asia, Bay of Bengal at the east coast of India, in the eastern, western, and northern Arabian Sea, and the Red Sea. And recently, it has become very abundant in the Gulf of Oman. Both the red and the green morphotypes actually overlap in their distribution in the eastern, northern, and western Arabian Sea, with a seasonal shift from green Noctiluca in the cooler, winter convective mixing high productivity season to red Noctiluca in the more oligotrophic, warmer summer season. These are the references I used for this report. This has been Paula Aliposa. Thank you for listening, and once again, good day.